Namaste dear all students. Today I am here to discuss about the DNA and RNA. Are you ready? Okay, all of you come front with your stationaries. This DNA and RNA, where do we study? Why we study? In previous class, we have discussed about the cell division, where we have talked about the chromosomes. Okay, then how these chromosomes are made up of? What are the components of those chromosomes? Yes, this DNA and RNA are the components of the chromosome. This DNA and RNA is in our unit 4, that is heredity. Okay, where it is in the which class? Yes, it is in the class 10, that is science and technology, obviously. Okay, what are DNA? DNA stand for? Yes, it is deoxyribose nucleic acid. Deoxyribonucleic acid, and another RNA is ribonucleic acid. Today's class is only about these two nucleic acid. Just discuss about something heredity. What is heredity? Or how does genetic characteristics transfer from one generation to another generation in organism? In previous class also we have discussed about the some characteristics of parents are transferred to the offspring. In some cases same to same characteristics are transferred. In some cases some characteristics are mixed up and those are transferred to the next generation. Then how these are transferred? What are the means of transformation, or how these are going from parents to offspring? With which mean? Just see here. Here the chromosomes present in human being are shown here. Yes, male and female. From the male, one set of chromosome, and from the female, another set of chromosome. That means in human being there are 46 chromosome. Out of those 46 chromosome, 23 from father and again 23 from mother. These are transferred to the child or the offspring. So while transferring those chromosomes, or those chromosomes are made up of DNA and some RNA also. So in this heredity we should know about the DNA and RNA, their structures, their functions in detail. That's why this heredity chapter is completely based on DNA and RNA, or our heredity depends on, or every organism's heredity depends on the DNA, especially because the whole chromosome is made up of DNA, where there are small genes are present. Those genes determine the characteristics of the organisms. Next, this girl is thinking. Always see around her there are goat is giving birth to its young one, those are kids, yeah, and this cow is giving its young one calf. Similarly, rat gives birth to its own young one. All are similar to their parents, even though some characteristics are different, and most of the characteristics are similar to their parents. How it is possible? What is the secret behind it? How this is transferred from one generation to another generation? That secret we reveal today. Okay? Dear students, once you see this one, this picture, this is your body, which is made up of cells. and inside the cell there is nucleus. Inside the nucleus there are chromosomes. Okay, these chromosomes are made up of DNA. In that DNA there are so many genes. In the DNA, the gene is the smallest piece of DNA which determine a single character. So we can say that DNA is the base of heredity. That means transferring characteristic from parents to offspring, and representing those characteristics in the offspring is done by this DNA. That's why whatever the structures we have, whatever the characteristic we show or other organisms show, all those depend on the composition of DNA or how the DNA is made up of. So this whole body is made up of cells, and this cell contain chromosome. That chromosome is made up of DNA and RNA. DNA contain many genes. So gene is the basic unit of transfer of heredity. So always we should remember that gene is the smallest piece of DNA. That's why DNA is very important in every organisms. Now what is DNA, RNA? We are saying nucleic acid. What is actually nucleic acid mean? What is the composition of it? This nucleic acid is the genetic material. It is a genetic material made of natural chemical substances. That means in the organism's body there are various type of chemical substances. Those chemical substances combine together and form the genetic material. That genetic material which is made up of such type of chemicals is known as nucleic acid. And these nucleic acids are of two types, already you know that. That is RNA and DNA, ribonucleic acid and DNA deoxyribonucleic acid. This depends on the sugar present on that molecule. That means DNA contain deoxyribose sugar, whereas RNA contain ribose sugar. Based on that, these nucleic acid are named as deoxyribonucleic acid DNA and ribonucleic acid RNA. How many types of nucleic acids he is thinking? Already you know that. There are two types of nucleic acid. One is DNA, another one is RNA. Their structures are also different. See, this DNA is double helically coiled. Double helically coiled. And RNA is single stranded. Here there are, in case of DNA, there are double strand. Two strands are helically coiled. Just like this. Okay. And RNA, it is a single strand. Now, what is DNA? What is the composition of it? How does it perform its function? Let's discuss. Dear students, we have seen this picture before also, is it? See, this is nucleus. Inside the nucleus, there are chromosomes. For example, if it is a honeybee or it is a queen bee, how many chromosomes are there? Yes, there are 32 chromosomes. And out of those 32 chromosomes, if we see a single chromosome, then in this single chromosome, if we see the structure, there are small chromatids are connected to a single point which is called centromere. Just we can see a chromosome. This is chromosome, structure of chromosome. If you see here, what you see, just there is a point where these chromatids are joined together. Okay, this point is called centromere. Okay, and these are the chromatids which are made up of DNA, and some part there is RNA also. This actually these are present inside the nucleus. Okay, inside the nucleus according to the species, just like human being they have 46, just like honeybee there are 32. Similarly, other organisms have their own number of chromosomes, and based on that this DNA role is also different. The characteristics are different in one organism to another organism based on the composition of the DNA. Now, what is there in this chromosome? Already you know DNA. In this DNA, we find smallest piece of DNA is known as gene. And what are the things present in it? See, there is written A, T, G, C, C, G. What are these things? This DNA is composed of different types of nitrogen bases. So, if we see this structure, where it is present, see inside the nucleus, okay, where? In the chromosome especially, but some DNA molecules are also present in the cytoplasm, in mitochondria, chloroplast, plastids, etc. also. Then how can you define DNA? Can you define, my dear students? Yes, long thread-like structure found inside the cell or even inside the nucleus, which represent the characteristic of certain organisms, or which are the genetic materials of the organism, that is called DNA. It carries genetic information. Already you know that it is a genetic material, so it carries the genetic information from one generation to another generation, or it shows the characteristic, or it represents the characteristics of the organism. For example, human beings have 2 eyes, 1 nose, mouth, 2 ears, like this. And in case of cow, they have tail and they used to walk with the 4 legs. Yeah, oh, that is determined by the chromosomes which contain DNA. Okay. Next, where DNA is located? The location of DNA is different in case of different organisms. Okay, in case of prokaryotic cell, this is a bacterial cell. Where it is present? It is in the cytoplasm. Freely it is present in the cytoplasm. Why it is so? You know that prokaryotic cell means it doesn't contain a definite nucleus. The nucleus is not present in the prokaryotic cell. Because of that, this DNA is freely present in the cytoplasm, which act as a genetic material. Next, in case of viruses, it is also genetic material. But in case of viruses, there is no cytoplasm. Nothing is present. Just this DNA is covered with a protein sheet called capsid. Inside the capsid, it is remain as the genetic material. Okay, the virus is a acellular organism. Acellular means cell is absent. There is no other cell components are present. It contains either DNA or RNA, which is covered with a protein coat called capsid. That's why this DNA is present in the viruses, just enclosed within the capsid. Next, in case of eukaryotic cell, it is present inside the nucleus, which we have already discussed. Now, deoxyribonucleic acid, DNA, how is it? Okay, it is composed of nucleotides, and the components of the nucleotides are nitrogen base, deoxyribose sugar, and phosphate group. There are 3 components especially. See, there are nitrogen bases, 4 types of nitrogen bases, which are called adenine, thymine, guanine, cytosine. And another one is there is sugar molecule, and this sugar molecule and phosphate molecule form the strand, this just outer ring, or this outer strand is made up of sugar and phosphate molecule, and inner part there is nitrogen bases. This DNA is double stranded. You can see here, this is one strand, another one is this strand, and these two strands are anti-parallel. They are anti-parallel and helically coiled, just like this. Okay, and these helically coiled structures are formed by the bonding between these nitrogen bases. Now, what are the nitrogen bases present in the DNA? These are adenine, guanine, cytosine and thymine. This is adenine, this is thymine, guanine and cytosine. These are the different 4 bases which are present in the DNA. How they are bonded? How these 4 nitrogen bases are located or present in the DNA? There is a base pairing. How they pair means complementary base pairing of these are, first one is adenine pair with the thymine. Adenine pair with thymine with double bond. You can see there is a hydrogen bonding between these 2 nitrogen bases. Okay, adenine binds with the thymine with 2 double bond, that there are 2 hydrogen bond. See, upper one and lower one. This double bond is seen in them. And next pair is guanine and cytosine. Guanine pair with cytosine with triple bond. See, this is guanine and this is cytosine. They are bonding with each other with the help of 3 hydrogen bonding. There are 3 hydrogen bonding. In this way, they are paired inside the DNA. This pairing ensures DNA stability and replication accuracy. In previous class also, we have learned about the DNA replication. Once again, nitrogen base pairing, how does it occur? Okay, how they are pairing? Say, suppose this is adenine and it is pairing with thymine. There is double bond. And this adenine will pair with, in this side, it will pair with a sugar molecule, which is deoxyribose sugar. And this also pair with another molecule, what is called, that is phosphate molecule. If it is up to this, one nitrogen base pair with one sugar molecule, then it is called nucleoside. If there is nitrogen base, sugar molecule and phosphate molecule, these 3 combinedly called nucleotide. Okay, in this way there are so many nucleotides are there. So many nucleotides, as for example, another one is, suppose guanine bind with cytosine. There is triple bond. And again it will bind with another sugar molecule. Here also this will bind with another sugar molecule. Again this will bind with phosphate molecule. In this way it will form a long chain, and but this is parallel to this, anti-parallel we say, and just they are twisted to form the helical model of DNA. And when there is pairing occurs, they are forming just A pair with T, C pair with G, that means cytosine with guanine. And when there is replication takes place, they will unwind. Just they unwind. Suppose this is the DNA which is going to replicate. Now at first it will unwind. Suppose there is A, T, G, here A pairing with T, T pairing with A, G pairing with C. Now after unwinding, what happens? It will take another T, and this T will take another A. like this, and here T will take A, and A will take T, G will take C. Now C will pair with another G, and now see, it is just forming the complementary copy. This is one DNA, this is another DNA. In this way, it will maintain the stability, or it will maintain the DNA stability in the new cell also. Next, what are nucleotide? Already we have discussed what is nucleotide. Where there is, just recall it, nitrogen base, deoxyribose sugar form nucleoside, and nitrogen base, deoxyribose sugar and phosphate molecule form nucleotide. These are the nucleotide and nucleoside. Each nucleotide have their own identity in a DNA molecule. Just see here, we can see here. These are the bonding between the ATCG, TA, GC, which we have already discussed. In each and organism, each and every organism have this unique sequence. That means, for example, in this we have seen after A there is C, T, G. In another organism or in another sequence, there may be just C, A, T, like that. That means it is changing. Based on that, the DNA fingerprinting, have you heard about this technique? When there is some crimes occur, what they do? They used to do DNA sequencing, where they can identify the persons who are related to that. That means while doing this, just they read about this sequencing of these nucleotides. Now, function of DNA. What are the functions of DNA? It carries hereditary information from one generation to another generation. Just like this dog is giving birth to the puppies. These puppies have the same genetic composition that of their parents, and contains genes. It contains gene. Already you know that gene is the smallest piece of DNA which represent a single character, and transmit genetic information during cell division. Already we have discussed in the previous class about the cell division, where we have seen how the characteristics are transferred from parents to daughter cells, and serves as a template for RNA synthesis. Here RNA synthesis also occur from the DNA, which is called transcription, and it enables protein synthesis. That means RNA is the main for the protein synthesis. That RNA is formed from the DNA. So DNA is also responsible for the protein synthesis. Next, gene. What is gene? See, already we have discussed gene is the smallest piece of DNA that represent the single character. Now, what is ribonucleic acid or RNA? How it is different from the DNA? See, it has only one strand, and it has uracil instead of thymine. Other nitrogen bases are similar. Cytosine, guanine, adenine. Instead of thymine, there is uracil. This RNA stands for the ribonucleic acid. It is a single-stranded polynucleotide. That means many nucleotides combine together and form the RNA, just like in the DNA. Where this ribonucleic acid is found or RNA is found? Generally, it is found in the cytoplasm. Just like in DNA, DNA is inside the nucleus, but this RNA is generally found in the cytoplasm. It is also present in the chromosome in a small amount, not like DNA. It serves as a genetic material in case of some viruses. RNA virus contain RNA as a genetic material. That means viruses have DNA or RNA as a genetic material, which contain DNA, those are called DNA virus, and which contain RNA, those are called RNA virus. In case of cells, see, you can see these are the RNA which are present in the eukaryotic cell, that is in the cytoplasm. Now, what is the composition of RNA? The RNA is composed of many nucleotides. Already you know that it is a polynucleotide. What it contain? It also contain nitrogen base, ribose sugar and phosphate group. Just like in DNA, in case of DNA there is deoxyribose sugar, here it is only ribose sugar, other composition are similar. The nitrogen bases which are present in the RNA are, say, uracil. Instead of thymine there is uracil and cytosine, adenine, guanine. These are the 4 nitrogen bases which are present in the RNA. How there is pairing occurs? In case of RNA, see what may be the, can you guess how they pair? Yes, there is no thymine, so adenine will pair with uracil. And there is double bond between uracil and adenine, just like in between adenine and thymine. Next, there is guanine will pair with cytosine with triple bond, just like in DNA. These RNA are important for formation of structure of the body. That means as they synthesize proteins, these proteins are the building block of the organism. So every living organism's body is made up of different kinds of proteins, carbohydrates, and those proteins are synthesized by the RNA. That's why it is very important for the development of the structure of the body. Now, there are different types of RNA. What are the types of RNA? There are 3 types of RNA. Mainly, those are messenger RNA. This is messenger RNA. Then that carries genetic information from DNA to ribosomes. That means this mRNA is formed from the DNA in the nucleus and it brings the information to the ribosome. You know that ribosome is the site of protein synthesis. That means ribosome is the cell organelle which synthesizes the protein. That's why we call it as a site of protein synthesis. That means this mRNA brings the information from DNA that is present in the nucleus to the ribosomes. And tRNA or transfer RNA. This transfer RNA brings amino acids to ribosome during protein synthesis. That means during protein synthesis, the amino acids, that means it smallest piece of that, you can say that after an analytical reduction of protein, amino acids are formed. The combination of many long chain of amino acid is called protein. Those amino acids are transferred by the tRNA. That's why it is called transfer RNA. Those amino acids which are present in the cytoplasm brought to the, that means those are brought to the ribosome with the help of transfer RNA. Dear students, next RNA is ribosomal RNA, which is present in the ribosomes, and it is the main which synthesize the protein. Okay, there are 3 RNA combinedly work for the protein synthesis, just like mRNA brings the message from the information from the DNA. tRNA brings the amino acids from the different parts of the cell to the ribosome, and ribosomal RNA present in the ribosome which helps in the perform the protein synthesis. In this way, these RNA are categorized into 3 types and their functions are also different, but in combinedly, that means these 3 combinedly helps in the protein synthesis. Now, how this protein synthesis occurs? Just we have learned about the 3 types of RNA. From that also it is somewhat clear. Yeah, main function of RNA is protein synthesis. Here it acts as an intermediary between the DNA and protein. Okay, that means DNA is performing some information, giving some information, and RNA is synthesizing the protein. That means in the ribosome there is protein synthesis occurs, which is with the help of DNA only. What information DNA sent to the ribosome, that based on that those amino acids are brought. So for example, this is one amino acid, another like this, they join and they form a long chain just like this, and in this way long chain is formed. Here I have shown only 3, but long chain it forms the amino acid chain, that is called protein, which is formed, that means which is done by these 3 types of RNA in the ribosomes. Just you can see here, you see tRNA is bringing these amino acids, mRNA brings the message from the nucleus, and here this rRNA or the ribosomal RNA is combining this protein, that means amino acids molecule in a chain that is called protein. In this way protein synthesis takes place. And in some cases, just like in virus, it gives genetic expression also. That means genetic characteristics are determined by the RNA. And in some cases it acts as a catalytic, that means catalyst, you know that. Catalyst means which help in the some biological chemical reaction. It helps to increase the rate of reaction. These are the functions of RNA. And from this picture, see here, you can differentiate between RNA and DNA. Based on our discussion, and observe this picture and write the differences between the DNA and RNA based on these structures, based on nitrogen base, based on ribose sugar or deoxyribose sugar. That means what type of sugar present in it, and also the pairing of, and now how many number of strands are present in the RNA and DNA. Based on that, just write the differences between the RNA and DNA and show to your teacher. Now we have discussed so many things about the DNA and RNA. Now let's check our knowledge through certain questions. First question is, which of the following is not a component of DNA nucleotide? We have discussed about the DNA nucleotide. There is deoxyribose sugar, nitrogen base, phosphate group, and ribose sugar. Which one is not the component of DNA nucleotide? Can you say? Yes, it is ribose sugar. Next question. If DNA transcription does not occur, which process will be affected? What is transcription? Yes, transcription means formation of RNA from the DNA. If RNA is not formed, which one will be affected? There are various life processes are shown here: photosynthesis, sexual reproduction, protein synthesis, and cell division. Which one is the correct option? Yes, it is protein synthesis. Next, some creative response question. Explain how the complementary base pairing in DNA contributes to its function as a carrier of genetic information. That means how this complementary base pairing of DNA, just like we have seen adenine, that means bind with a thymine, and guanine with a cytosine. When they form the replicates, how they are forming and how they are helping in the transfer of genetic information, how those arrangement of nucleotides help in the transfer of genetic information. Write about those things and show to your teacher. Next one is compare and contrast the structures and function of DNA and RNA, which we have discussed in our class. Based on that, write just what are the similarities, what are the dissimilarities. Just compare and write the similarities and dissimilarities about the DNA and RNA, and all those work you have to do in your note copy and show to your science teacher. Today we have discussed about the various topics based on DNA and RNA. I hope you have learned a lot. Thank you and have a great day. Bye-bye.